This is an international project looking at sequestration of carbon in the seabed in estuaries, and what we are going to be doing is planting two very specific types of tea bag. One is rooibos and hibiscus and the other is green sencha, but they're very specific because they're in plastic containing tea bags which are no longer made. So you've got two types of tea bag. Each box has got enough for 2 quadrats, and you need half of that box and half of that box for your quadrats. I think I know more or less where they are because I only put them out last week. Today we are planting out in the seagrass area and in the mud, and yesterday we planted all 4 soil types. And then the data we'll use for restoration projects, looking at whether we sequester carbon. And all you need to do is put this in, move it out, and then as you move it out, you put that in, and then put that back down, and put your label on top. That should be fine. So it's very quick.
It's not really, and then it just does it going down there. I feel it, the whole thing as well. It's quite good.
It's alright. I'm just kind of
pushing it down.
Yeah, slotting it into place.
It'd be awesome to come out and be part of the community and give back in some sort of way and contribute to a cool, interesting, novel citizen science project.
I think there's a lot of climate potential that we can unlock through blue carbon. I really like the idea of a worldwide experiment where lots of countries are getting on board, and we'll hopefully get a really huge, really useful data set from it.
Projects like this gets you involved and you get to be around the people who actually know about it in depth.
Green or red?
Oh, sorry, those are green.
In just day-to-day life, I think, being conscious of climate change and the way we look after our environment and the changing climate, I think that speaks to me quite in a personal manner. And I think that's why these type of projects, I'm drawn to them and I'm quite keen to be involved. It's nice to be able to be part of something bigger than my day-to-day.
It's great getting my hands dirty, getting my boots dirty.
In the estuarine area, we like to have the plastic in the tea bag because it means it's not going to decompose. And we want to be able to dig them back up again in 3 months and find the same tea in the tea bag as when we put it in there. That's our ultimate goal. If the tea hasn't been decomposed, then that's an indication that the carbon's been sequestered under the seabed. Whereas if it's being decomposed by microbial communities mainly, that during that process the carbon is released back out into the atmosphere. Right, so 40. Okay, so this is 40. So this is a thicker seagrass. So who's got 4041? The seagrass has a lot of different jobs. It can be used as a nursery for lots of different species. Some species feed on it. It can provide some protection from erosion and the wave action when you've got a large amount of it. So it has a number of jobs that it does in the estuary. The salt marsh tends to be more on the edges, and so that provides more protection against erosion for the coastal edges. And again, they both provide habitat for lots of species, so that's why we want to restore them, because we've lost an awful lot of the seagrass and the salt marsh.
So you get lots of different species of these ones in New Zealand.
Is it that colour, or is that
I'm not sure when they're big, but when they're small, they're transparent.
People don't really know about blue carbon, or when they look at estuaries, they don't really think that they have some sort of climate value. And so what I'm thinking is that if people understand that, then they would be more inclined to want to protect and enhance those habitats.
All this is doing is looking at whether it's likely carbon is sequestered under the seabed. So the seagrass and salt marsh are really good at sequestering carbon, and also because of the large area that you have usually that's either mud or has nothing growing in it, it also sequesters carbon to a lower level, but it's quite high because of the volume. So it will be good to see what difference there is in New Zealand, and those results are going to be uploaded to the international site as well. And so we'll be able to look at how our sites compare to international sites, and also the different variabilities of the temperature, humidity, and things like that we'll be comparing as well with our results. When we dig them up again after they've been under the soil for 3 months, is we might take some soil samples and analyse those later, looking at microbial communities as well to see if they're different under the different plant or soil types.